I'd like to start with a huge thank you to all of you for coming along this afternoon. It's been a tremendous gathering, and also a huge thanks, I think, from us all to Michelle for the amount of effort and energy and dynamism that she's brought to this project, and I certainly picked that up this afternoon from her opening talk onwards. So thank you very much for coming along. What I'll do is try and expand on some of Sally's work, outlining some of the basic science, the fundamental discovery science that goes on at the Oxford Parkinson's Disease Centre. So, as we know, this year is the 200th anniversary of the publication of James Parkinson's paper on Parkinson's. So I thought I should go and read it. I won't embarrass any of the researchers here by asking them to put up their hands if they have read it, but I went back and read it, and some stuff in there is absolutely fantastic. First of all, he would have been a great friend of Michelle and of this cohort. So he wrote, "The disease is of long duration. To connect, therefore, the symptoms which occur in its later stages, which those as mark its commencement, requires a continuance of observation of the same case, or at least a correct history of its symptoms, even for several years," which is a fantastic description, I think, of the discovery cohort. He also picked up many of the subtle symptoms around a disordered state of the stomach and bowels, and noted that the sleep becomes much disturbed. He had perhaps an unwarranted faith in the therapies of the day when he wrote, "The intelligent will never fail to avail themselves of any opportunity of making trial of the influence of mercury." He also recommended bloodletting from the neck, which I do not either recommend. But he was an absolute optimist in what could be done in Parkinson's when he wrote in 1817, "There appears to be sufficient reason for hoping that some remedial process may ere long be discovered by which at least the progress of the disease may be stopped." That was 200 years ago. We share his optimism and we're working hard at it, but it's difficult going, and I'll try and explain a bit of the work that we're doing in our centre. So it's been known for a long time in academia and pharmaceutical industry that drug discovery has not been working, especially for some of these difficult neurological diseases of the brain. So drug discovery is a lengthy process. I've outlined some of the aspects of it there, from discovery through the preclinical phase, where you're working in cell or animal models, through to phase 1, 2, and 3, where you're testing out the potential therapies in patients, through to FDA approval and use. And our work would be right down the left-hand end, understanding more about what goes wrong in the disorder and the disease and how we can attempt to treat it. So it's lengthy and it's expensive. So on average, it takes about $1 billion to get from the left to the right to drive a new therapy through to market. And it's getting more difficult. So you can see here in purple, that's the bar graph that represents the amount of money that's been put into research and development worldwide. And this flat yellow graph is the new drugs, the new medical entities that are coming out. So it's getting harder and harder. So our own work is understanding disease mechanisms, particularly around the stem cell-based models that we have been able to grow with collaboration with Sally, because you have been kind enough to donate skin biopsies for us. So we work with this stem cell technology, these induced pluripotent stem cells that Sally described. We call them a disease in a dish. They're able to recapitulate some of the cellular features of Parkinson's. So, and thanks due to Michelle collecting the samples and Sally's effort in generating the stem cell lines, we have the largest stem cell collection of Parkinson's stem cells in Europe, over 175 lines so far. We've worked to identify key cellular pathologies that go wrong in the Parkinson's neurons, and we hope this will as well as support the research, but also able us to inform some of the clinical diagnoses. Can we tell a different form of the disease from the cells that we study, and would that back up some of Michelle's clinical findings? And we've used these to initiate screens for new drugs and targets. And it's been a very collaborative effort to make these dopamine neurons from the skin biopsies that you kindly provide, through to reprogramming to stem cells in Sally's lab, through to generating these dopamine neurons in my laboratory. And the neurons that we make have many of the features that you'd like to see in a neuron. They look like neurons on the dish. They send out these long electrical connections known as axons across the dish. And they stain positive with many of the neuronal proteins, such as tyrosine hydroxylase, TH, which is the defining marker of a dopamine neuron. These neurons can make dopamine, and they respond to L-dopa by making more dopamine. They have electrical activity. They fire, they fire electrical signals across the dish without stimulation, just as the neurons in the substantia nigra do. And they also, as I'll come back to in a few minutes, have a very typical form of calcium signaling. They take in and pump out calcium rhythmically, very distinct neuronal pattern that matures over time to this very rhythmic frequency of calcium signaling. So we can make good neurons. And we've been working hard at studying them, again, as a collaborative effort. You can see Michelle and Sally are both on this paper here. The writing's a bit small, but earlier last year we were able to publish our findings showing that when we work with these neurons derived from Parkinson's patients who carry mutations in a gene called GBA, we can identify some things that are going wrong in those neurons that aren't going wrong in the control healthy neurons. For example, they suffer from a level of protein stress, because the cells cannot process excess protein correctly. And that leads to a protein stress in this famous protein in Parkinson's called alpha-synuclein. These cells can't recycle it effectively, so they release it. And there's a developing theory that Parkinson's may in part be caused by a spread of a protein across the brain, such as alpha-synuclein. And maybe that's the case. Maybe these neurons are releasing the alpha-synuclein more than they would otherwise. So this was one of our recent papers. We've published others, including fundamental characterization there as to how you make these neurons. We've looked at gene expression profiling in these neurons, I'll come back to. We've started to think, like Sally, around the glial cells, not the neuronal cells, but cells such as astrocytes or microglia that help and support these neurons. So there's three aspects that we bring to this target and drug discovery through our cellular models of Parkinson's in our center. We have a fantastic collection of the human neurons from Parkinson's patients. We've identified cellular pathologies, things that are going wrong with the cell. We have a new drug discovery institute funded by Alzheimer's Research UK and started in Oxford about 2 years ago. It's got Alzheimer's in the title, but it's not just for Alzheimer's at all. It's for age-related neurodegenerative diseases. And we're supporting them and providing them with projects in Parkinson's. And we're now starting to obtain several drug libraries from publicly available libraries that you can get, or from very bespoke collections, for example, one that arrived 2 weeks ago from AstraZeneca for us to screen. And so this work requires a big investment in equipment and infrastructure as well. So about 2 years ago, Sally and I worked together to obtain a large funding from Medical Research Council, which we also obtained money from the university, and occupied, if you like, took over the lab upstairs and turned it into a laboratory for doing cell-based screens to look for potential drugs and targets, filled it with some very exciting equipment, and we're now getting it into use, and it's now working. This is our favorite piece of equipment, is a very sensitive microscope that can scan across 384 wells at a time. It cost about as much as a semi-detached house in Summertown, but it did come with a free cake. So they delivered the cake, and we've enjoyed the cake. We're now using that. And as you can see, some of the robotics that's required to handle hundreds of samples in parallel in these tiny well plates are now up and running, and we're getting some of these projects well underway. So what are we doing? Well, those of you who've followed our program and worked with us and come to discussions and talks before will know that we have a theme for the clinical work, a theme for the cellular work, and a theme for the animals work. So as we move into the last 2.5 years of OPDC2, we are very much collapsing the efforts between the cell and the animal work to bring it together, to bring across 5 sort of therapeutically potential areas that span across the stem cell neurons to the rodent models. And I'll go through each of them very briefly. So first of all, we're developing ideas of repurposing compounds that have used in one disorder, and we might be able to repurpose them for use in Parkinson's. Different ways of clearing out toxic alpha-synuclein. Different ways of boosting lysosomal function. Different ways of working in the area Sally talked about, trying to dampen down bad neuroinflammation, and also think about how we might help the mitochondria, the energy power plant of the cell that packs up in Parkinson's. So briefly, what are we doing in those 5 areas? So the first is where we come back to calcium I mentioned a while ago, and that is that neurons, dopamine neurons in substantia nigra, have a very particular way in which they handle calcium. And calcium, which enters the neuron through calcium channels, promotes neuronal death in Parkinson's. And there's one particular type of calcium channel called the L-type calcium channel, which is very targetable. And we intend to investigate this using a family of drugs called gabapentinoids. This is an area of research which Stephanie Cragg, a neuropharmacologist in the centre, is very interested in. So the idea is that in a neuron before gabapentin treatment, a lot of calcium could be fluxing into the neuron, and that causes stress. With gabapentin treatment, we could block some of those calcium channels and reduce the calcium stress in these neurons. So could this family of drugs be used to prevent or slow the progression of Parkinson's? Michelle also has enthusiasm for the development of these treatments because they're already used in a number of clinical conditions. They're used, for example, in a number of neurological conditions such as neuropathic pain or epilepsy. They could be quickly repurposed to treat Parkinson's. They're well tolerated by patients. We know they're used for some Parkinson's patients who have sleep problems. And in a recent survey by Parkinson's UK, people cited gabapentinoids as drugs they were taking, which also helped them with some of their symptoms. So this is a good example for repurposing, and what Stephanie will be doing is to take some of the gabapentinoid drugs and testing them in our Parkinson's mice, and if successful, we might then be able to move them into the clinic. Another project uses not the mouse models but the stem cell models that we have. So we published earlier this year a study where we made dopamine neurons from Parkinson's patients who carry a mutation in a gene called LRRK2, and we compared the gene expression in those neurons with control neurons and found 168 genes in these purified neurons that were expressed differently between disease and control. We then compared that to an online database which records how certain drugs change the expression of genes. And we reasoned that if we could find a drug that had an equal and opposite effect on gene expression to the disease, we might be onto a potential therapy. And in this totally unbiased blind approach, we identified a compound called clearquinol, which has a long clinical history and previous use, often as an anti-malarial, but it's also been tested in a number of neurodegenerative diseases, reaching phase 1 and 2 in Parkinson's and Alzheimer's. So in our totally blind approach, this was something we identified, so we've started working further with clearquinol to try and understand it. The next approach is to try and clear out the toxic alpha-synuclein. We've heard a bit about alpha-synuclein from some of the other speakers. It's a normal natural protein, we all have it. But as Parkinson's develops, it accumulates in clumps and builds up into oligomers, which are sort of small clumps, and then fibrils, and then ultimately into Lewy bodies. And whether these Lewy bodies are toxic, whether they're killing the neuron, or whether they're protective as the neuron tries to package away the synuclein and survive, we don't know. So work by Pete McGill and Paul Dodson has done some world's first experiments in this, in which they've adapted a previously used technique to generate alpha-synuclein fibrils inside the mice inside the brains of mice. That's been done before. But what they were then able to do is to stick their electrodes into the substantia nigra of a mouse, which is very tiny, and record how the neurons behave when the Lewy bodies are there. That's something, of course, you can't do in people, but you can do it in this mouse model. They found that early on the Lewy bodies weren't killing the neurons, but they made the neurons change their electrical firing pattern. And so here is a normal neuron ticking away, bap, 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 and here's the neuron that's carrying the Lewy body. So even when the Lewy body isn't killing the neuron, it's making it behave incorrectly. So that's a first, and we're now working to understand how we can improve that. So how are we going to improve it? How are we going to clear out toxic alpha-synuclein? Well, we've heard work that Michelle is doing with Biogen using antibodies in a potential clinical trial. Here's another piece of work that we're doing in the lab. How can we unpick toxic alpha-synuclein aggregates? Well, we've teamed up with one laboratory on the west coast of States, from Los Angeles and from a group in Paris, to use a compound that's known as a molecular tweezer. It's able to go around and pick apart the alpha-synuclein aggregates. In this picture here, I hope you can see in our mouse models, you've got lots of red dots, lots of alpha-synuclein aggregates that have accumulated. We use our molecular tweezer, which you can sort of see even looks like a tweezer, and we're starting to show that we can pick apart those aggregates in the mouse models. The question is that beneficial? Would, for example, it return the neuronal function that we saw in the previous slide to normal? Another way to harness the immune system's benefit in Parkinson's is not to use monoclonal antibodies, but actually to generate a vaccine. So we've worked with experts at the Jenner Institute in Oxford who make vaccines for different diseases, and they helped us make a vaccine against alpha-synuclein, which we showed raised the correct antibodies. We published this last month to show that we can generate antibodies using this vaccine in the mice. We now need to understand whether this is going to be potentially therapeutic. We come to lysosomes. So this is the cell's recycling bin. We heard from Sally how important lysosomes are in recycling protein. I want to turn the lights down again. So we study lysosomes, which are the structure here. So there's a structure called an autophagosome which collects stuff around the cell that needs to be recycled, and it delivers it to the lysosome, which is the recycling plant that I've shown here as a recycling bin in a yellow box. And we're able to grow neurons, this time from our Parkinson's rats, in the laboratory and identify these formation of these lysosomes and see how efficiently they're working. So we can show in a normal, non-transgenic rat a formation of these lysosomes. We can work with cells taken from transgenic rats carrying a LRRK2 mutation, and they have a deficit in lysosome formation. Treat these neurons with clearquinol, which we found in a completely separate study in the human neurons, and we're able to correct this lysosome dysfunction. Neuroinflammation. Is this a potential therapeutic approach for Parkinson's disease? We heard from Sally that she has the stem cell derived astrocytes and microglia, and you can't watch a good video too many times. So there's Sally's video showing the microglia surveying locally in the dish. We've been working with our mouse models as well, and we found that if we look at the astrocytes and the microglia in our mouse models when they're young, 3 months old, about an 18-year-old teenager equivalent, we can see they don't have many astrocytes in green, and they have these normal microglia that look like this. If we wait till the mice get to 24 months, which is about a 75-year-old person, or 12 months as well, accumulation of astrocytes, and these microglia are no longer the happy form that Sally showed us, but they've started to become activated. So we've accumulated in these older mice astrocytes and activated microglia. Can we start to clear them out? We know that as these mice age, they accumulate the alpha-synuclein fibrils. If we unpick the fibrils, will we correct the glial dysfunction? And finally, the mitochondria. So mitochondria are the powerhouse of a cell generating energy, and mitochondrial failure has long been suspected to occur in Parkinson's. And this is a figure from our stem cell derived neurons, and every red dot is an alpha-synuclein fibril, and we can show an accumulation of alpha-synuclein fibrils in the patient neurons, and as these fibrils accumulate, the mitochondria fail. This is looking at mitochondrial function in blue in control neurons and in red in the patient neurons, and as the alpha-synuclein accumulates, the mitochondria fail. We can measure these mitochondria, the dimensions of them. We show they get more fragmented, they start to fall apart in the cells, and we know they start to fall apart in the mice as well. So it's bringing together the cellular models and the mouse models, and I know people with Parkinson's often want us to collaborate and interact, and we're developing a collaboration between our group and Sheffield, who've taken some of the discovery cohort patient cells and have worked with their models to further understand mitochondrial dysfunction. So this, or versions of it, are one of my favourite slides, and I know Michelle's as well, because it emphasises what we've managed to build here as a national international centre. We have collaborations with a new centre in Luxembourg. We work with many groups across the United Kingdom and across Europe. But down in the bottom left-hand corner, you'll see a box you might not have expected, which is that we are getting increasing approaches from major pharma partners, pharma and industry, who have essentially got stumped by Parkinson's. They want the better models, they want a new understanding, and they approach us to help them develop new understanding of what goes wrong in the cell biology and novel therapeutics. And we must be doing something right if we're having a lot of interest and companies knocking on our door to help fund our work. So just to finish off, where does this all go? Well, Michelle and you guys put together a fantastic cohort, which enables us to do the biomarker studying, the imaging, and some of our genetic studies. The neurons that we're making, the glia that Sally's making, are bringing this to a better understanding of the cellular changes that happen and the ability to look for drugs that may correct these cell changes. The rodents I described to you about, it's only when you work with living mice or rats you can record from Lewy body neurons deep inside the brain to help us understand the disease, develop new therapies that we can ultimately bring back to the clinic. So this work was all done by people in my group and beyond, the Target and Drug Discovery team at OPDC, together with some of the best collaborators you could ever ask for, and thank you very much for your attention.